Yeah, thank you all for being here today. It's exciting to be part of this awesome lineup of speakers. And my talk here is focused on our recent work to develop a more generalized understanding of motor cortical control of skilled movements. And I'll tell you what I mean by that in just a moment. I'm a bit of an outlier here in this session because my work is focused in primates, but my hope is that we can use part of what we're finding now to directly guide the development of experiments that leverage new capabilities in humans, like what Uli and Matt talked about earlier. I'm using a probably straightforward and pedestrian definition of interpretable latent spaces throughout my talk. Here, I'll use that to mean anything where we can algorithmically recover population-level signals, where we can describe in words how variability along certain dimensions relates to anything that we care about, like behavior. So, drawing inspiration from the success of visual neuroscience, the field of motor control has sought a canonical computation using constrained tasks like 2D planar reaching, which led to the early theory that neurons in motor cortex use population vectors of neurons with cosyne tuning to signal an upcoming movement. And then, of course, a little over 10 years ago now, Mark Churchland and Krishna Shenoy challenged this model with their discovery of rotational dynamics during reaching. And while they were pretty careful to describe their results as pertaining to, being specific to how the motor cortex controls reaching movements, many in the field were quick to assume that rotations could serve as a kind of general basis for generating a really wide range of different behaviors. And I think it's worth popping up a level and considering everything else that the motor system has to do and is capable of. And in general, I think the main theme here is that it may not be the right question to ask, what is the single canonical computation or a unifying principle for how the motor cortex generates all possible movements. And it's almost trite to point out examples of skilled motor behaviors, but I'm going to do that anyway, using Jackie Robinson, who is obviously a groundbreaking athlete and Baseball Hall of Famer. And we're going to use him to underscore two separate points. First, there's an enormous gap between our understanding of motor cortex in controlling basic planar reaching movements and the types of computations that we know the brain has to be performing in order to generate real-world and skilled movements like hitting a fastball. And it's completely unclear how our current models of reaching would generalize to these more complex feedback-guided skills. Second, he wasn't just amazing at baseball, but as a college student, he also played varsity basketball, football, and track, and he was actually also a tennis champion. So it's also completely unclear how the motor system can learn and separately control an enormous repertoire of completely different skills and keep these skills separated without interference. And addressing that full complexity is likely not really possible experimentally in the near term, but our goal should be to at least create a model or a framework that can plausibly scale to address the range of computations that we know the system has to be performing just based on natural behaviors that we can observe. So I'll start this talk by describing a recent experiment to study the control of multiple skilled movements in different feedback control contexts. And I'll then walk through a simple model and offer a bit of speculation for how I'm now thinking about a little differently about the general problem of how the motor cortex controls movements. And then at the end, very briefly, I'll describe some experimental directions that I think we should collectively pursue that leverage the cognitive flexibility to study motor control in human patients using some of the new methods that were described previously. All right, so just as a high-level overview, the motor system is responsible for transforming high-level inputs that specify goals and task constraints and knowledge of the body dynamics in the world into dynamic signals that control roughly 650 muscles to produce behavior, and then obviously use sensory feedback to guide and control the unfolding movements. And I'm, of course, not the first person to point this out, but as a field, we have yet to really grapple with and design experiments that kind of address the degree of computational sophistication that these very simple block diagrams imply. So to study how the motor system is capable of learning and expressing multiple different skills, but with an approachable level of complexity, I've adapted a version of the Pac-Man task that Naja Marshall originally developed, and Ilham Mamamatsu, who spoke a couple minutes ago in a parallel session, helped considerably with all aspects of this work. And in this task, the monkey presses on a handle to generate isometric forces to control the vertical position of a cursor, while a path of dots scrolls in from the right edge of the screen. That allows us to specify arbitrary and precise sequences of muscle forces that the monkey has to produce. And in this task, we've created a second context where I invert the gain between the force and the cursor movement. And here in the reversed blue, the blue context, the reverse gain context, we flip both the visual stimulus and the gain between the force and the cursor movement. And so importantly, the monkey's always pushing forward on the handle, but in one context that moves the cursor up, and in the other context that moves the Pac-Man cursor down. And while it might seem like a little bit of a stretch to call this a new skill, we've directly altered how the monkey needs to respond to visual feedback to control his movements, and it actually takes around 6 to 8 weeks to train this. So it's relatively challenging for the monkey to learn how to do this. So we'll consider two general models for how the monkey's brain might solve the challenge of performing this particular task. One possibility is that areas upstream of the motor cortex, like the parietal cortex, could simply flip the visual error signal. And the motor cortex then performs the same computation using the same inputs that it would receive in the two contexts. And I'll call this the unified control model. And I would argue that virtually all of, most previous work in motor control kind of implicitly assumes this type of model, where the motor cortex is kind of the last stop before the output and is doing the same thing to control a similar movement. Alternatively, though, the brain might be able to learn and store a separate independent controller for this negative gain context. And under that model, upstream areas of the brain would send inputs to select different sets of dynamics that are appropriate for each different control context. And something like this has to be taking place somewhere in the brain. But since the motor cortex is the last stop before the periphery, and directly generates the muscle control signals, it's pretty natural to think that activity there primarily encodes the output, while upstream areas of the brain, like the parietal cortex, could handle a very simple sign flip computation. And perfectly honestly, before we started this, we really expected to see this kind of unified control strategy and thought that we would have to go looking to other brain areas to see where these other interesting computations would take place. So that was our prior. Here's a video of the monkey performing the Pac-Man task. When the path is red, he's pushing forward to move the cursor up from the bottom of the screen. And then the path turns blue, the cursor flips to the top of the screen. He's still pushing forward. He's always going to be pushing forward, but in this context, that moves the cursor in the opposite direction. And the monkey's behavior is precise and repeatable across many trials. He's able to do this really well. It does take a number of months of training, but after that training, we can see that the forces are well aligned by design across these two contexts for this matched set of conditions. And we can also vary the amplitude and frequency across different sets of conditions to create, here are 3 matched pairs of different force profiles. And we have other condition types as well, like steps and ramps and things like that, but we'll start with these for simplicity. And you can see it's not perfect, but they are pretty well aligned across the different contexts. So now we can make some predictions for what we expect to observe in motor cortex under either the unified or the task-dependent control strategy. So under this first unified control model, with the flipped visual inputs, under this model, the motor cortex is simply generating output, so we would expect the neural activity should be identical across the two contexts. And we also would expect to not be able to decode context information from the population response. And finally, we would expect that any response to visual information or perturbations would primarily still just be encoding outputs. But alternatively, if the brain is using something more like this task-dependent control model, then we would expect that there should be distinct neural activity across these two contexts. That we potentially could decode context information, potentially even before the movement begins, once that cue is present. And lastly, that any perturbation response to visual inputs would include a mixture of all sorts of different signals, including visual inputs, internal computations used to generate the appropriate outputs, and then the output signals themselves. So during this task, we recorded from thousands of neurons in the premotor and primary motor cortex using the NeuroPixels NHP probes. So these are new probes that allow us to resolve signals from 200 to 400 different neurons, sometimes even more on an individual probe, and it's very straightforward to record from multiple probes simultaneously. And that immediately solves two major challenges for us in electrophysiology. First, we can record a huge number of neurons, and it also provides access to large populations of neurons that are deeper in the brain, which we're actually not leveraging for this particular study, but you can read more about in our paper. So it's essential to acknowledge the really large team of people who have been working to make these possible and who have all been instrumental in developing and testing the probes. And our preprint is now online as of a couple of weeks ago, and anyone interested in purchasing the probes should be able to do so within a couple of days, actually. They're just becoming available right now. So we've designed and built an insertion system that allows for precise trajectory planning, dense packing of multiple probes, and safe operation. We've had zero probes broken in the brain, which is nice. So here's a rendering of that approach using 8 probes or 3,072 simultaneous recording channels. And we've recently begun testing this system, and it's pretty, so looks a little weird, kind of crazy, but it's actually really straightforward to use this. Like, I didn't think that would be the laugh line, but no, it kind of just works. And so that's like a incredible new capability where this is what that looks like in practice. And while that scale and capability are fun and interesting, the exciting part is obviously the new science that we can do with that. So with that, let's look at one neuron. So here's a good neuron. The red and the blue traces here are the firing rate of this neuron across the red and the blue context, and this is like a nice intuitive. It strongly correlates with the output force in the bottom right here in the two contexts, and it feels like a good 4C neuron, like what motor cortex should be doing in the task. But then we also see other neurons that are a bit weirder like this. So this is a neuron that reliably responds only in the red context and remains almost entirely silent in the blue context. There's also neurons with the opposite response. So we have neurons that respond robustly in blue but remain silent in red, so that's kind of weird. And then we have other neurons where there's responses in both contexts but with pretty different phase relationships across the two. So I've shown you a few neurons, but we want to see if these are typical responses or whether they cluster into different discrete response types. And to address that, we are going to visualize whether there's any clustering. So we just apply PCA to the concatenated trial average data and then visualize the clustering of PC weights using t-SNE. And at that visual analysis, we don't see any evidence of clustering, and that's also true if we apply analytical methods like E-pairs analysis. And so the lack of clustering suggests that the context-dependent signaling in this task isn't a property of different discrete subpopulations, but is kind of a distributed population-level phenomenon. So we want a quantitative metric, though, for analyzing the dissimilarity in neural activity between the red and the blue contexts. And so to calibrate our expectations, I ran a second set of experiments here where we cue the monkey to generate explicitly different levels of force, but all in the same direction under the same gain. Same gain context. So in the high force condition, the monkey generates around 66% more force than in the low force condition. And then we use demixed principal component analysis, or DPCA, to quantify how much of the neural population variance we can attribute to the different force levels. And as many of you are probably familiar, DPCA looks for dimensions of the neural population activity that contain variability that can be attributed to differences between conditions and kind of factorizes that out. So Using that approach, we can factorize the population variance into, dare I say, interpretable components that are dictated by the structure imposed by our task. And when we applied DPCA to the force scaling task, we find a dimension here that captures variability that's independent of the different force levels and varies with time but not target condition. And we also find a dimension where force levels do separate, but this dimension captures a lot less of the total population variance, so a lot less than the 40% in this first component. And these are just two dimensions, but if we look at the full factorization of variance in different components, we see that the force scaling accounts for only a total of 13% of the total variance, and it's kind of distributed across all of these different components here. And that's pretty counterintuitive because we might imagine that the motor output should be the dominant signal in the population response in motor cortex, but it's actually relatively small. When we apply the same analysis to data from the context switching task, we see a kind of remarkably different picture here. Context makes up almost 40% of the total variance. We still see this time-varying, condition-independent dimension 1, but in a second dimension, we see a really clear separation between activity across the different contexts, and there's also a number of different components here with a large amount of variance that's attributable to context. And then comparing these results directly, we again see this kind of curious picture where the amount of variance is remarkably larger in the context tasks, where the output is actually matched and very similar relative to the force scaling task, where we've explicitly asked the monkey to do something very different. And we absolutely didn't expect this to be the case ahead of time, and found this result pretty surprising at first. But the variance only tells us a portion of the story. So let's visualize the neural state space directly. So here I've applied PCA to the trial average neural activity, and we're plotting trajectories in just the top 3 principal components, so nothing too exotic. And the neural activity oscillates, as we might expect, and occupies this kind of ring in neural state space. And then we can also plot all of the neural trajectories for the positive gain conditions simultaneously. So despite the different amplitudes and frequencies, the trajectories for all of these conditions for the positive gain trials kind of live in a very similar region of this space. And then if we add in the negative gain conditions, we'll see that the positive and negative gain trials kind of occupy very different regions of the space, and that the set of and they kind of separate out by these different rings. And so that's despite the differences in the actual kinematics of the movement, we see this large context separation, and everything kind of clusters based on context. So now let's take a look at when this context sensitivity emerges over the course of the trial. And in particular, we're interested in whether context signals are present kind of independent of the actual motor generation process. So here we're just, again, simple analysis. We're using a linear SVM with cross-validation to classify trials by context. And we see that when the monkey receives this context cue, well before, several seconds before before he actually starts moving, the signals in motor cortex immediately diverge and reflect the context information. So that robustly decodable signal for context seems to translate activity into the appropriate portion of the neural state space to generate the subsequent movement in a context-appropriate way. That's nicely consistent with our second context-dependent control model, but was initially quite surprising to us. So returning to our predictions here, we've now seen in a couple different ways that neural activity driving the output behaviors in the positive and negative gain context is quite different. And finally, we want to check this, whether this last prediction is true, whether we see any portions of the population response that contain signals related to the inputs and internal computations in addition to the outputs. So to understand the role played by inputs, I introduced a new task variant with a slowly ramping forces, and on a fraction of trials, we're going to jump the cursor. So here the monkey is controlling the Pac-Man, the cursor jumps, and he's able to rapidly correct and continue. So they can do this immediately. This isn't something that needs to be learned. They essentially do this first trial. And the monkey responds with appropriate corrective forces to counter the error that we've introduced by positive and negative gain perturbations, cursor perturbations. So this is the force that the monkey generates in response. And that's nicely mirrored across both contexts. So now we want to ask whether there's dimensions of the neural population activity that kind of track these visual inputs that are separate from the dimensions that drive the actual outputs. So again, we're going to use DPCA on the perturbation data, and here we find that the first component, which in this data captures over half of the total variance, is again attributable to context. So the red and the orange trials up at the top here correspond to the trial average activity for perturbed positive gain trials, and the blue trials at the bottom are perturbed negative gain trials. And then we also find a second component, which captures 12.7% of the total variance, that looks a lot like a force output. So here, the positive and negative gain conditions that require more force in response to the cursor perturbation cluster at the top, and the conditions that require less force in response to the perturbation cluster at the bottom. And third, we also find a dimension that captures neural variance that we can attribute to the cursor jump visual input. So the two upwards perturbations cluster here, and then the two downwards perturbations cluster here. So this dimension kind of looks a lot more like a visual input dimension, which is pretty interesting. So now we've, all of the data that I've shown you is consistent with this second model where motor cortex is implementing a separate and kind of independent controller for each of the two contexts, and it's not acting as a single unified controller, not performing one unified computation across these two control strategies, even though the output behavior is completely identical. All right, so in the last few minutes, let's think about how this might actually work at a kind of mechanistic level. So I'm going to step into a more speculative domain and kind of sketch out some schematics for how we think this might be working. So a key concept of the dynamical systems perspective is simply that the dynamics govern how the neural state evolves from one time point to the next. So in an unperturbed trial, we have a neural trajectory that evolves, or a neural state that evolves along a normal neural trajectory. And then when we add a visual perturbation, this kind of kicks the neural state off from this unperturbed trajectory. And then the local dynamics in the network govern how the neural state evolves from one time point to the next. So we can schematically plot these local dynamics as a flow field, and here this is just in 2D space, but the concept generalizes to a much higher dimensional space. And we can use this concept to create a controller that translates inputs into appropriate outputs. And so in this case, the visual perturbation pushes the neural state along an input dimension, and then the neural state will then evolve along a dimension that produces the appropriate, in this case, lower force output. And the issue with this is that this mechanism obviously doesn't work if we need to use a single set of local dynamics to respond differently across the two different contexts. So an obvious and easy mechanism for solving this task is to just spread things out in neural state space so we can have different sets of local dynamics to perform a different computation on the same inputs. So now, in the blue case, when the perturbation comes in, that allows the neural state to evolve in a way that generates more force in response to the perturbation. And then in the red case, when the input comes in, the neural dynamics will allow for the appropriately lower force response to the perturbation. So this is only obviously possible when we've spread things out in neural space using these two separate flow fields. And that requires separated trajectories for these two different contexts, which is consistent with everything that we observe in the data, but is sort of a speculative extension from that. So jumping even further into speculation, in theory, a really high-dimensional neural state space provides a pretty large capacity for different sets of learned motor skills or motor memories, which is kind of a reverse cursive dimensionality. What I think is a kind of cool idea here is that individual skills could be confined to different regions of a high-dimensional space, but that being able to leverage this high-dimensional space gives a pretty large capacity for different sets of new motor memories. And as new skills are learned, potentially the motor system could use different parts of this space to learn distinct dynamics and allow for different computations to be isolated and performed in different sets of this space. This idea could at least plausibly explain how we're able to learn a pretty vast repertoire of different motor skills, keep them distinct and isolated, and then index into different skills as necessary based on different low-dimensional contextual cues from other brain areas. Obviously, we want a more complete map of this space. This is just kind of a hand drawing. And we want to gain interpretability for how the variability in different dimensions of the space lead to different types of performance across different tasks. So testing a greater range of behaviors and more intricate tasks could benefit considerably from the cognitive flexibility and intelligence of humans. And so we're working on ways of running analogous experiments in clinical populations soon. For our central conclusions, the reason why I think these results are exciting to me is that they suggest kind of a new perspective for the type of computational flexibility that I think the motor cortex could be using to perform a range of different skilled behaviors. So different skilled movements here require different responses to input, which require different local dynamics to transform inputs into appropriate outputs. And then different dynamics require a system to kind of spread things out and use different parts of this neural space to be distinct, even when outputs are identical. And then more broadly, I think that it's highly likely that motor cortex is not performing a single canonical computation, but is instead a really flexible and highly capable machine that kind of spools up different dynamics that are appropriate for different tasks. And so many of the conclusions that we draw from our observations are likely not highly generalizable beyond the specifics of the tasks that we're studying. So then, in summary, I told you about our one recent project today, leveraging the high-density recording technologies and focused on understanding different visual motor skills and how they're controlled by motor cortex. Presented this sort of speculative general framework, providing a plausible but not very compact mechanism for how the motor cortex could potentially learn and control a wide range of different skills. But I think really fully testing these ideas will require considerably more complex behaviors that leverage the types of recordings in human patients that are just starting to become possible now, where we can provide verbal instructions, test a wider range of contexts, and a much wider range of different motor skills. Lastly, I'd like to thank my numerous collaborators, mentors Mark Churchland, Mike Shadlin, and Daniel Wolpert, as well as Tim Harris and Barun Dutta, and the rest of the team at IMEC and the rest of the NeuroPixels NHP Development Consortium. And then lastly, my workshop co-organizer, Sabera. So with that, if we have any time, I'll take any questions.